Today we're going to talk about targeted microanalysis, or TMA. And SciAps offers the only instrument that's able to do this type of analysis. It's a screening method that allows you to focus on a very small surface area. Generally, our LIBS instrument can focus on a spot size about 50 microns wide and about 10 microns deep. So this is the type of analysis that you need a scanning electron microscope or an ICP to do. But now you can do it with a simple screening tool like SciAps LIBS. There are three aspects to TMA. The first aspect is quantitative analysis. And you can do this by testing the carbon on small samples like screws. In this photo right here, you can also test inclusions and look at components to see the makeup of a very fine-tuned area for analysis. Okay, the second aspect of TMA is qualitative analysis. This includes 2D mapping, and it also includes just focusing on an individual spot or location that you want to identify what elements may be present in a sample. A good example of this is in electronics testing. Usually in a clean room environment, they want to test the surface and see what kind of contamination has got on the sample. So in these cases, they'll use something like Element Pro. Test just the surface. Just take one shot, one location, and try to identify the contamination on the surface. A good example of this is in the jewelry industry, where they'll often use beryllium coating to make the stone look more clear and more valuable to a potential buyer. So in order to identify this, you can test the outer reaches of the stone to identify whether or not there's beryllium and it's been treated. Another aspect of the qualitative analysis is using Geochem Pro. And in Geochem Pro, you can do mapping of the surface of any stone or geological sample that you would like to look at. This is often done in gold mining. And what they do is they'll look for a gold vein. First of all, identify the gold in the sample, whether or not it's gold, maybe pyrite, maybe it's not neither. And then they will look at what is surrounding the gold. So if the gold is surrounded by calcium and carbon, basically calcium carbonate, what happens is that the carbon in the calcium carbonate eats up the cyanide, which is used to pull out the gold. And so therefore, that particular gold vein would not be economical to mine. So they use this as an identification tool for looking at the mineability of certain veins. And also with lithium mining, they'll often look at pegmatite, whether or not the pegmatite is lithium bearing or not lithium bearing. The third aspect to TMA is semi-quantitative analysis. And this can be done by adjusting the settings on the LIBS. So with the third aspect of TMA, you can adjust the settings to the instrument. You can go to acquisition settings, you can go to the factory acquisition settings, and you can edit the roster positions to just one position. Then you can adjust the cleaning shots, take it from 2 to 0, so we're just testing the surface. Then you adjust, so we're only taking one data shot. And then the next step is to adjust the data pulse averaging to 1. So that allows you just to take a very simple test, just one location, one data shot, and that's it. One of the clearest examples of semi-quantitative analysis using TMA is testing individual components of lithium-ion batteries. Each cell, the user or recycler will take the instrument, they'll just use one data shot and analyze just the surface. And what they'll get out of it is the rough idea of the lithium concentration and also be able to identify, is it a lithium iron phosphate battery? Is it a lithium nickel manganese? Is it lithium nickel cobalt manganese? They'll get a good idea of what the battery is and how to sort the battery for later processing. So here is an individual layer of a lithium ion battery. And what the recyclers are going to do is they're going to take this component and they're going to test just the individual layer using TMA. So we've built a curve for black mass, but now what we're doing is taking that individual curve that we use cleaning shots and data pulses for, and we're going to convert it into just one data shot and get the results. So we're going to test this sample. It's a very quick test, and you'll get the result right away, as seen here. You can also grab many components and take repeat shots. So we'll take this sample and we'll test it on multiple locations to look at the consistency of results that we're getting with this type of analysis. So we'll start off by taking one shot, and then we'll repeat that shot again and do it at a different location and see the consistency between the two results that we're getting. So that just gives you a brief example of all the applications that you can do with TMA. The applications are absolutely limitless as far as what you can do with the instrument. It depends on what elements you want to see, how you want to focus the analysis, whether it's just surface. Do you want to just take one shot on the surface? Do you want to actually produce more cleaning shots and drill into the surface and see what's below a surface coating? The options are limitless. And with SciAps LIBS, it allows you to see elements that you weren't able to see before with traditional XRF analysis or other types of analysis. And really the biggest advantage to using LIBS is that you don't need all this preparation to do it. It's very simple. Just grab, point, and shoot. And you can save yourself a lot of time and energy with your analysis programs.